We've gotten pretty good at treating tumors that stay put. But cancer has a dark side, a truly terrifying ability to move, to spread, and to come back when you least expect it. And that, right there, is its deadliest trick.
It's the big question, isn't it? The one that keeps doctors and scientists up at night. How can we be winning the battle against a tumor in one spot, but then lose the war when it suddenly appears somewhere else? What is happening inside that cancer cell that allows it to go on this fatal road trip?
And this is why it's so incredibly important. This one sentence really just lays it all out. We're not just talking about a scientific curiosity. Understanding how cancer spreads is the central challenge. Metastasis, that process of spreading, it isn't a side plot in the story of cancer. It is the main story.
To solve a puzzle this massive, you really need a completely new way of looking at it. And sometimes that new perspective comes from a journey that's not just about science, but is also deeply personal.
So let me introduce you to Dr. Sundari Mani. He was the son of farmers from a tiny town in southern India. Get this, he was the very first person from his entire hometown to ever get a PhD. His journey took him all the way to MIT, to one of the best labs in the world, where he came face to face with that huge question: how do cancer cells learn to spread? And right there, he found a clue, a fundamental biological process that would change everything we thought we knew. And here it is. This was the breakthrough moment, published in the journal Cell. Now, the title sounds super technical, I know, but what it's really saying is that cancer cells perform a kind of magic trick, a shape-shifting act that gives them the powers of a stem cell. Dr. Mani figured out that when a cancer cell gets ready to spread, it doesn't just start crawling away. Nope, it transforms into something totally different, something tougher, more resilient, and way, way more dangerous.
So, what is this shape-shifting process? Well, it has a name: the epithelial mesenchymal transition, or EMT for short. And honestly, if you can understand EMT, you can understand the very heart of metastasis.
Okay, let's break this down. Most of the cells in our body are what you'd call epithelial. Think of them like bricks in a wall. They're stuck together, they're organized, they have a job to do, and they stay put. But during EMT, some of these cells go rogue. They transform into mesenchymal cells. They let go of their neighbors, change their shape, and basically become these lone wanderers with the power to move and invade new places.
And this image shows it perfectly. Look on the left. You see that neat, orderly sheet of cells all lined up. Now look at the right. They're breaking free, becoming individuals. The analogy of putting on a disguise is just perfect. The cancer cell sheds its old identity so it can survive the incredibly dangerous journey through the bloodstream, find a new place to live, and start a whole new tumor. And this is the real kicker. It's not just about movement. Dr. Mani's discovery showed that this transformation gives the cancer cell a whole new set of deadly superpowers. Suddenly, it's resistant to chemotherapy. It can survive floating around in our blood. And, most importantly, it gets the stem cell-like power to start a whole new tumor all by itself. This one process explains both how cancer spreads and why it comes back.
So if EMT is the process, what flips the switch? What tells the cell to go rogue? Well, the answer is deep inside the cell's command center, and it involves a family of master control proteins that are like double-edged swords.
Right, let's meet the key players, the FOX family of transcription factors. These are basically proteins that tell your genes when to turn on and when to turn off. But as you can see from this table, they're not simple good guys or bad guys. Look at FOXA1. It can help turn on cancer genes, but in some situations, it actually stops that dangerous EMT process. Or FOXM1, which makes cancer resistant to drugs, versus FOXO3, which can literally tell a cell to self-destruct. It's this incredible complexity that makes cancer so tricky to fight.
And some of these FOX proteins have a really special skill. They're called pioneering factors. Why? Because they can do something most other proteins can't. Imagine your DNA is a book, but most of it is locked tight. These proteins are like molecular locksmiths. They can find those lock sections, pick the lock, and pry the DNA open. This allows all sorts of other cancer-causing instructions to be read for the first time. It's a complete reprogramming of the cell from the inside out.
Okay, so all this molecular biology is fascinating, right? But this is the moment where the story goes from the lab bench to the patient's bedside. This is where we see how this knowledge can help us decode why some cancers are so much deadlier than others.
Okay, take a look at this heat map. All you need to know is that bright red means highly active. And what you're looking at is the genetic signature for that whole EMT shape-shifting process. See where it's glowing red hot?
That's in metaplastic and claudin-low breast cancers. And those aren't just random names. They are two of the absolute worst, most aggressive, and hardest to treat types of breast cancer. The link is just staring us in the face.
And if that heat map was the clue, this right here is the proof. These are survival curves from real patients. The red line shows patients who had low levels of one of these EMT switches, a protein called FOXC1. The blue line is for patients with high levels, and the result is just devastatingly clear. The more that shape-shifting switch is turned on, the lower the chance of survival. This isn't just about cells in a dish, this has life-or-death consequences.
But here's the thing. With this deep, deep understanding comes real hope. Because if we know what the master switches are, well, we can start figuring out how to turn them off.
So this research gives us a roadmap for a smarter way to treat cancer. Step 1, you analyze a person's tumor to figure out exactly which of these FOX factors, which of these switches, is causing the problem. Step 2, you use a targeted drug, a smart drug, to shut down that specific switch, essentially taking away the cancer's ability to shapeshift. And finally, step 3, you hit that now vulnerable cancer with chemo to finish the job. It's a 1-2 punch designed to stop the spread and kill the source.
And that leaves us with this incredible, tantalizing question. The whole journey, from a small farming village in India all the way to the cutting edge of molecular biology, has shown us that cancer's deadliest trick might be controlled by just a handful of switches. And the challenge for us now is to figure out how to flip them off. And maybe, just maybe, stop cancer dead in its tracks.